Today we are going to understand the eyeball. First of all, there are three layers or three coats: the outer fibrous, middle vascular, inner nervous. Fibrous includes sclera, cornea. In the diagram, the yellow part and the blue part seen outermost are the two fibrous coat. The middle vascular includes the three structures we'll see in the diagram. First is the choroid, the red part. Then there is the brownish part of the ciliary body and iris covering the lens in front. These three form the middle portion. And the third, which is there, it is said to be the innermost nervous layer, which is the retina, the yellowmost portion which we can see inside. Then the light which is there first passes through the cornea, then enters the aqueous humor which is present, then crosses the lens, then enters the vitreous body. So this entire process is known as the way in which light enters our eyes, and these all structures form the refractory media. We can see the optic nerve entering from behind and an optic disc in the diagram. The dura mater which is labeled continues ahead to form the sclera. There are suspensory ligament holding the lens. In the anterior portion you can see the aqueous humor divided into anterior and posterior chamber. There is a sinus venous serrae. You can see sclerae which is present. There are conjunctiva present. Sagittal section means cutting the eye lens in two half and watching it from one side. Now we'll look at the outer fibrous coat. Okay, first the sclera which is there, it is divided by the sclerocorneal junction from the cornea. The sclera is thinnest at the anterior portion where there are recti muscle inserted, and where the thickest or the weakest part of sclera is where the optic nerve enters. Lamina cribosa is the structure of sclera which has numerous perforations for the entry of the fibrous processes of the optic nerves. Okay, apart from that, let's look at sclera. Sclera forms the posterior 5/6 part of the eyeball. Anterior 1/6 forms the cornea. Okay, it is dense fibrous tissue. Apart from the basic things, we saw the thickest and thinnest regions of it. The thinnest portion is 6 mm behind the sclerocorneal junction. It is also known as limbus. Then there is Tennant's capsule which covers the sclera, but anteriorly the sclera is covered by the conjunctiva, due to which the sclera appears white when we look through the eyes of any person. It forms the white portion of the eyeball. Inner portion of the sclera is a bit brown in color, and if we look at the place between the sclera and choroid, there is a perichoroidal space, and in the perichoroidal space there is a lamina, that is lamina fusca, and this is also having another name, that is suprachoroidal lamina. Okay, I told you limbus is the another name for sclerocorneal junction. Now the canal which is present, canal of Schlem, is present over there, which is going to have the function of draining the aqueous humor. Okay, and where is this present? It is present in the deep portion of the limbus or the sclerocorneal junction. And this canal of Schlem drains the aqueous humor from the portion anteriorly and moves it into the artery which is present. And we look at the artery diagram of the artery in the diagram ahead. Okay, so here is the artery which we were talking about, that is anterior ciliary artery, which is marked now. There is vena verticose and long posterior ciliary artery present in structure of eyeball which are piercing. Physiological cup is present behind. The two space between the vitreous humor is hyaloid canal. I showed two axes, vertical axis and optic axis. Then sinus venosus or sclerae I showed you, iris, cornea, ciliary body, suspensory ligaments. All these labelings I'm just showing you again to be verified with the diagram much more. Now these structures which I showed you in this diagram are important for you to understand how these structures are piercing the eyeball. Aqueous humor is cleared by all, the axis of eye is cleared, then visual axis and optic axis were the two axes which I had shown. Okay, dura mater, I told you dura mater is which continues to form the sclera. Apart from that, sclera is pierced by 4 structures. Okay, the 4 structures include the one which is shown first is the optic nerve at the center, and in the optic nerve there is presence of artery and a vein, which I'll talk about later on. Optic nerve is present infero-medial with respect to the posterior pole of the eyeball. That structure is short posterior ciliary artery and 4 vena porticosae. These are 4 veins, or they are also given another name, that is choroid veins. Okay, and the fourth structure which is piercing is the ciliary artery. The anterior ciliary artery is the fourth structure piercing the sclera. These 4 structures is very important to be remembered. Okay, in the sclera, basic things you understood what you need to write. This was the sagittal section and this was the structures piercing. Sclera is avascular, but the presence of sclera and conjunctiva, which is the covering of sclera, there is a region known as episclera between the two of them, which is vascular in nature. In sclera, you need to write all the structures piercing, the 4 structures, and the covering of sclera, how it is thinnest and thickest in different regions, and it is forming the fibrous outer layer. Cornea is transparent in nature. It forms the anterior 1/6, and it is said to be more convex with respect to the sclera. Okay, because we know it's present in front of the lens. It is separated from the iris. Okay, look at this diagram over here. The iris is the brown colored portion and cornea is the outermost. It's separated by the aqueous humor. Specifically, the anterior chamber of the aqueous humor separates the two of them. Look at the diagram. This is the aqueous humor, anterior and posterior part of aqueous humor. Now if we move ahead, cornea is also said to be avascular in nature, just like the sclera, and the blood supply is not present here, so the nourishment is supplied is by the lymph, and the lacrimal fluid nourishes the cornea. Now what are the branches which supply cornea? The nerves. Okay, it is the branch of ophthalmic nerve which supplies it, and short ciliary nerve. So these 2 nerves are important. The only reception which is present over here is pain, which is quite well felt. So you need to remember these all points in cornea which is highlighted. Now we look at the histological structure of the cornea. Okay, we saw the outermost layer in the histologies. First we'll see the epithelium present, that is stratified non-keratinized squamous epithelium. The second layer, which is that is Bowman's capsule present, or Bowman's membrane, sorry, not capsule. This we'll call as anterior elastic lamina. Okay, there is a posterior elastic lamina which we'll discuss a bit later. This portion which is present in between is substantia propria, and the part of substantia propria at the lower portion is known as the predecim membrane, a predeciments layer. Okay, here there is absence of keratocytes. Okay, that's why it's a given different name, and there is lamella of collagen which is present here. Then the layer which is present at the lowermost portion, there are 2 layers. This is Descemet's membrane, and lastly there is posterior epithelium made up of squamous mesothelium. Okay, now we saw the certain layers in the histology: squamous epithelium, Bowman's membrane, substantia propria, predeciments layer, deciments layer, and squamous again. So just we are sandwiching it. So this is histology of cornea. Okay, this is important. The slide is going to be remembered with all the labelings. Substantia propria is thick in nature. Okay, now after moving ahead, we saw how there was different ways of understanding the fibrous outer layer which was covered through the diagrams. Now cornea which is there, it can be grafted because it is avascular. If dust enters into the eye, we can feel pain. Then bulbar conjunctiva which is present, it is vascular and its inflammation leads to conjunctivitis. And palpebral conjunctiva helps us to determine the hemoglobin level. So these are all the clinical anatomy points which are important. So the portion which is present below the eyeball is palpebral, and the portion which is present over the we can say cornea, which is that is the scleral portion over the sclera, is known as the bulbar conjunctiva. So difference between bulbar and palpebral, I think it's clear. Palpebral below the eyelids and bulbar over the sclera. Next is the anteroposterior diameter. Shape and curvature of the eyeball is quite important, because if there is any defect, it could lead to myopia or hypermetropia. Now we look at the next, that is the middle layer. middle coat which was vascular, it included three things: choroid, ciliary body and iris. First we'll look at choroid. We can see the red portion, okay, moving ahead the brown, and then the iris which is covering the anterior part of the lens. Iris is going to manage the diameter, okay, amount of light entering the lens is managed by the iris. Choroid is pigmented and it has 4 layers. We'll see choroid in detail. Ciliary body, you can see over there the structures which are present. If you try to look at it, ciliary body in a cross section, you can see it is surrounding the eye lens in a rounded fashion with the iris present just beside the lens. Now this is sclera and cornea present. I have shown a very close section, ciliary body, and we can see this is presence of the iris, okay. The ciliary body continues as iris which divides the lens, sees the lens which is divided into posterior and anterior chambers of aqueous humor. Canal of Schelm is seen over here, and this is also sphincter pupillae is also seen over here. Ciliary muscles are present in the ciliary body. This is suspensory ligament, and we can see there is vitreous humor behind the lens. See the same diagram over here. The lens, we have cut the eye in half, okay, so that's why we are seeing in half manner. This is the iris again, this is the ciliary process and ciliary body. The ciliary muscles are present here. Ciliary muscles also we'll discuss later. So this was cutting the lens in two halves, okay, exactly two halves, and looking at one side of it. Choroid is a thin layer which is pigmented in nature, and if we look at choroid, it continues ahead as ciliary body, okay. So choroid is not shown over here in the diagram, but ciliary body which is there is started only after choroid gets over. So choroid gets over at ora serrata. Ora serrata is a point, okay, on the, we could say, the retina of the eyeball, and over there the retina where it ends, ora serrata it is known as, from there the ciliary body is going to start. So basically that's the point of end of choroid. Just this much you need to remember. And choroid is going to start from which point? You can imagine choroid is going to start from the region where the optic nerve enters the eye, right? So that's just the boundary you need to know. Choroid is anteriorly bounded by ora serrata, and posteriorly it is bounded by the optic nerve, or the entrance of the optic nerve is where it starts. Now sclera and choroid were separated by a space, okay, which was known as perichoroidal membrane was present, and sclerochoroidal lamina was present. Now if we look at the region where sclera and choroid are joined together, it is quite loose, so sclera and choroid can be separated, but choroid has retina beneath it, right, and choroid and retina are held tight together, very firmly held, so it is quite difficult to separate the two of them. So mostly when there is any separation, it occurs only between choroid and sclera. The 4 layers of choroid are, first is sclerochoroidal lamina which we saw just now. Then there is a vascular lamina which is present where there are blood vessels. Then the third is choriocapillary lamina. Here there are loads of capillaries present, blood capillaries. And last is the Bruch membrane, okay. It is also given a different name which needs to be remembered. Now ciliary body which is there is the second structure which continues after the ora serrata, right? We saw ora serrata where it ends, from there the ciliary body is going to start, and we can see the structure of ciliary body. It is said to be triangular in shape. It's not so obvious, but we know it's just a bit triangular in shape. Then posteriorly ciliary body which is there is said to be pars plana, that is plane in nature. There are no bumps present posteriorly, but anteriorly there are folds present. You can see the pars plicata type of nature because there are 70 ciliary processes. So this was about ciliary body. Posterior surface plane, anterior surface there are processes. Next is what is ciliary zonule, and we are going to talk about the ciliary muscles which are present. Now ciliary zonule which is there, it is a structure which is present on the posterior surface of the ciliary body, okay. and this structure which I've highlighted in yellow, it is said to be a thick lining. But who is lining the ciliary body to form a ciliary zonium? It is a lining of the vitreous, and it's on the posterior surface. Okay, it's the lining of the vitreous membrane. This lining forming on the posterior surface of the ciliary body is known as ciliary zonium, and posteriorly it lines the hyaloid fossa, and anteriorly it forms a suspensory ligament. Okay, ciliary muscles are circular, longitudinal, and radial. Longitudinal are also known as meridional muscles. Ciliary muscles are said to be unstriped in nature, that is, they are having no striations. You can see this in the diagram, the circular and meridional fibers. Meridional are said to be present in this triangular format, which are extending from anterior region to posterior. So basically they arise from, you can see the sclerocorneal junction from where they are arising. Okay, these muscles are arising from the sclerocorneal junction, and just behind sclerocorneal junction the region is known as scleral spur. You can see the diagram, scleral spur, just near the sclerocorneal junction, or the limbus. So this is the place from where we can see the longitudinal fibers or meridional fibers arise, and they move backwards to the suprachoroidal lamina. Circular fibers are combining themselves along with the radial fibers to arrange themselves as an aggregate, okay, in front of the longitudinal fibers. Now they are present in anterior part of the ciliary body. Thus you got a muscle covered. So ciliary process, first you'll write it's triangular in shape, right? Apart from that, posterior surface is plane and anterior is smooth. Okay, one thing, these ciliary muscles are parasympathetically controlled. Okay, now if we move ahead, we'll look at branches of ophthalmic artery. We'll look at the iris, which is the third part of the middle portion of the eyeball. Anterior part of the uveal tract. We said that the middle part of the eyeball is known as uveal tract, which include choroid, ciliary body, and iris. Iris adjusts the light which is entering the pupil by acting as a diaphragm. Diaphragm is like a structure which is going to allow only specific amount of light. Okay, otherwise we can say it's like a door like structure. It divides the aqueous chamber into anterior and posterior chamber. Okay, the anterior chamber and posterior chamber division of aqueous chamber is done by iris. The peripheral margin which is there of the iris forms the ciliary body. You can see in the diagram, the iris is present in front of the lens. This is the structure, and between the iris and cornea the angle is known as IC angle. Okay, iridocorneal angle, and the anterior free margin, peripheral margin is attached. Okay, peripheral margin is attached to ciliary body, and free margin is present on the lens we saw covering the lens. Not it's exactly on the lens, it just rests on the lens. This is the iridocorneal angle which separates iris from the cornea. Now if we move ahead, the next point is that the anterior surface which is there is said to be covered. The anterior surface of iris is covered by the mesothelium, while the posterior surface which is there, it is said to be deeply pigmented cells which are covering it are deeply pigmented cells. Now the iris which is there, the bulk of the iris is formed by certain structures, that is the stroma, which includes the blood vessels along with the loose connective tissue. Now major and minor arterial circles are formed in the iris. Now we'll see the diagram of the divisions of ophthalmic artery in later on. The color of the iris is due to the pigmented cells present in the loose connective tissue we saw just now, which forms the bulk of the iris. And if it is absent, if these pigment cells are absent, the iris appears blue in color. Now pupillae, dilator, these are the two structures which we'll see. Before that, we'll see the ophthalmic artery. This is the artery entering from behind. Okay, this is the central artery which is present. The central artery of retina, I told you that optic nerve has an artery entering within it with a vein. So this is the artery which I was talking about. Now from the side you can see there are two arteries entering. The one which is going straight up till the lens, this is known as the long ciliary artery. Okay, and the other one which joins it from the sides forms a circle. The two circles are formed. The one which is present within the iris, that circle is known as minor, and the outside one is known as the major. So I told you that was a long posterior ciliary artery, and this one towards the inside is short posterior ciliary artery. The artery which joined to form the major circle was the artery accompanying the rectus muscle. Okay. The major circle goes into the iris part to form the minor circle, the minor arterial circle. That was the optic nerve, and the space between the optic nerve and arachnoid matter is subarachnoid space. And arachnoid matter has surrounded itself with dura mater. So this diagram makes it clarified that how the circles are formed. This circle, if we look at it from front, it is forming the entire circumference like this. This is the, in the center you can see this is the minor circle, and outside there's the major circle. So if we look at it from the front view, it forms the entire circle, but from the side view that we saw just now, we could only see the side view of the circle that we made, just a small circle, but actually it forms the whole circle. In this diagram you can see the ciliary zonule and ora serrata. Ora serrata, I told you it was the point where the retina stops, right? And the ciliary zonule was formed when there was the covering of vitreous humor or vitreous body on the posterior surface of the ciliary process or ciliary body. Now if we move ahead, we look at the clinical anatomy of certain points. Now if certain object is kept at infinity, okay, at a very far of distance, then what happens, the ciliary muscles present are going to relax, suspensorial ligaments are going to tighten themselves, and lens is going to flatten. Similarly, if a substance is present quite close to each other, ciliary muscles contract, suspensory ligament relax, lens becomes rounded. When we squint our eyes, that time we look at a place beyond the object. We look at a place, I would say, beside the object, not at the object, due to squinting. Now the innermost layer, that is the retina, is said to be the delicate nervous layer. That is, there are neurons present over here. It is covered by hyaloid membrane on the inner surface. Okay, inner surface of retina is covered by hyaloid membrane, and there are pigmented cells which are present on the outer surface. Now in the retina we saw the optic disc. Okay, this is the optic disc, the junction present over here. This is the iris which is present, ciliary body, this is the lens. So just I'm drawing a rough diagram to show the optic disc in the retina. Now retina, which is there, it is divided into, first is there is a region known as optic region, ciliary region, and iridial region. Optic region is the nervous region, and the rest two are non-nervous, that is, there are no neurons present. Now look at this, the retina which is shown over here, and the optic nerve. Retina is in yellow and optic nerve is present behind it. Ora serrata, see the point where the retina ends. Now we can see the optic region. Optic region, we saw it starts from ora serrata and it ends behind till the optic nerve or optic disc. Okay, look at the diagram again by going a bit behind if you want to clarify. Okay, if we move ahead, optic disc, we finish. Now the non-nervous portion is the portion beyond the ora serrata, that is in front of the ora serrata, and the ciliary and iridial region of the retina contains two layers of epithelium. Okay, two layers of epithelium are present in ciliary and iridial region of retina. Peripheral cups are region where there is no rods, no cones. It is said to be a blind spot in nature. And we have another region which is a depression known as macula lutea, which is a yellow color region present. Macula lutea, which is it has a central fovea where only cones are concentrated. Okay, it is avascular in nature. Okay, I told you it's yellow in color and it has only cones, and it is the region of maximum acuity. Okay, look at the diagram in detail. Now rods which are there, they are said to have visual purple and they show scotopic vision, while cones show photopic vision. Now there are 10 layers of retina which we need to learn. First is the pigment cell layer, rods and cones, outer limiting, nuclear, plexiform, inner nuclear, inner plexiform, ganglion cell layer, optic nerve fiber, and inner limiting fibers. So I have just a short trick to remember this. In order to remember this, just make a mnemonic and cover it up. The 10 layers of retina are quite important. First is PCR, okay? PCR L NP square GOL. So L you can consider that it PCR limits, but you say no problem. And GOL is some person's name, no problem Golu or something like that. So PCR is a limiting thing, but no problem Golu. So you can just apply it over there. It follows the 10 layer sequence. Now aqueous humor is a clear fluid. We saw it was divided by iris into anterior and posterior aqueous chambers, and into the posterior chamber there is certain structure via capillaries, we can say. In ciliary processes, aqueous humor is said to enter. Now into the anterior chamber, okay, aqueous humor enters via the pupil. Drainage of aqueous humor takes by the iridocorneal angle into the anterior ciliary veins with the help of canal of Schelm. Okay, we saw the canal of Schelm earlier. That's how aqueous humor is drained. If aqueous humor drainage is blocked or there is certain kind of issue in the drainage system, then it leads to glaucoma. Okay, it's a severe condition in which there is a certain disturbance because aqueous humor is accumulated in the anterior portion. It is rich in glucose, ascorbic acid, and amino acid. Aqueous humor is rich in these things. Now it is used to nourish the avascular tissues. Okay, aqueous humor is used to nourish the avascular tissues present. See, retina, we saw it can be detached from a structure which is present, so we can prevent it by using a silicone band and a sponge. Now lens, which is there, it is transparent, biconvex, circular outline is present, and it is consisting of 1 diameter. Okay, axis which is there, it is said to form poles, and there is equator present in the lens. The lens is going to vary in dioptric power. Power is 1 by focal length, that is dioptric power is inversely related to focal length. Posteriorly, it is said to be more convex. The lens is said to be posteriorly more convex. Anteriorly, it's relatively flat. Okay. Anterior, the lens capsule is the first layer. Second layer is the epithelial cells, which includes a cubical central region with elongated peripheral cells. Apart from that, these fibers contain nucleus. Okay, these nucleus are said to be firm. And there's an anterior posterior pole in the lens, obviously. Suspensory ligaments are holding the lens, and they form zonule of ZINN. Okay, they are responsible for keeping the lens in proper position and the tension is maintained. If there is left third nerve paralysis, it leads to pupil dilation along with the cornea moving downwards. If both, not both, I would say, there is partial ptosis along with meiosis, known as Horner syndrome. And finally, brainstem damage, then both the eyes are affected. Lenses are generally opaque in color. If lenses are not generally opaque, but if they begin to form opaque, it leads to cataract and it's seen during aging. Ophthalmoscope is the object used to see the eyeball. Okay. Vitreous body is said to be colorless. It is jelly-like in nature. We saw it is present behind the aqueous humor. It is enclosed in hyaloid membrane, and the homogeneous membrane which is present is the hyaloid membrane. Vitreous body extends from the optic disc behind till the ora serrata.